this contains everything you need to know about cells and transport in one diagram. So I will guide you on how you need to go through this. First, let's look into the definition. Cells are made up of nucleus and cytoplasm. Cells can be divided into two types, that are prokaryotes and eukaryotes. Prokaryotes are basically like bacteria, while eukaryotes are plant and animal cells. When you look at the structure of cells, there are three main things, that are plasma membrane, cytoplasm, and the nucleus. While cytoplasm has more components, that are endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, and peroxisomes. So let's look into the plasma membrane. Plasma membrane contains lipid, proteins, and carbohydrates. Lipids are made up of phospholipids, glycolipids, and cholesterol. They help to form the phospholipid bilayer. And proteins contain two types, that are integral and peripheral proteins. While integral proteins act as enzymes and transport carriers, while peripheral proteins function as enzymes and receptors. Carbohydrates are formed by proteins and lipids. And if they are proteins, they're called glycoproteins, and if they're lipids, they're called glycolipids. They also help to act as a receptor and sometimes antibodies as well. Just for a note, everything mentioned in green color is the function. The functions of the plasma membrane is they help to control substances that come in and out of the cell, and it also protects the cytoplasm and maintains the cell shape and size. Now let's look into cytoplasm. So cytoplasm contains ER, and ER can be divided into two types, that is RER and SER. RER is rough endoplasmic reticulum, while SER is smooth endoplasmic reticulum. The reason it's called rough is because there are ribosomes attached to the surface. And the main function of RER is biosynthesis of proteins, while SER is synthesis of steroid hormones and cholesterol. Golgi apparatus acts as the main transporter in cytoplasm, and they help to transport the synthesized proteins across the cell, and they are also involved in synthesis of lysosomes and peroxisomes. Lysosomes are formed from Golgi, and they contain hydrolytic enzymes that helps in destruction of bacteria and other foreign bodies. They also help to remove excessive secretory products and help in removal of unwanted cells. Peroxisomes are formed by division of pre-existing peroxisomes, and they contain peroxidases and catalases, and they also help in metabolism of peroxide, and they also play a major role in beta oxidation of fatty acids. So mitochondria is basically the powerhouse of the cell. So they act as a power plant as they produce ATP, and there are two main membranes, outer and inner membrane. The outer membrane is permeable for metabolites, while the inner membrane is the site for oxidative phosphorylation. And mitochondria is mainly involved in citric acid cycle, fatty acid oxidation, and it also helps in cellular respiration, heat generation, and initiating apoptosis. So nucleus is present in eukaryotes. They contain DNA and RNA, and they help in replication and transcription. That's its main function. So let's now look into membrane transport. Membrane transport has two types, one is active transport and passive transport. Passive transport can be divided into two types, that are simple diffusion and facilitated diffusion. So passive transport contains simple diffusion. Simple diffusion is when lipid molecules pass through along the concentration gradient, while facilitated is same again along the concentration gradient, but this time with the help of a carrier protein, and which I mentioned before, integral proteins. Active transport is movement against the concentration gradient. Since it's going against, they require energy. So active transport has two types, that is primary and secondary. Primary is when they use energy directly from hydrolysis of ATP, while secondary is when they use energy from electrochemical gradient. Secondary has two types. One is co-transport and counter-transport. Co-transport is when substances move simultaneously in the same direction, while counter-transport is when they move in the opposite directions, like one forward, one back, likewise. So when you look at transport of macromolecules, there are two main types. One is endocytosis and exocytosis. Endo means eating, while exo is spitting. So endocytosis has two types. One is called pinocytosis and phagocytosis. Endocytosis basically means taking up large molecules, while pinocytosis means taking up cellular fluids, so which is also known as cellular drinking. So this can be molecules like proteins, polysaccharides and polynucleotides, while phagocytosis takes in large particles like cells, bacteria, or virus. Exocytosis, on the other hand, is when, let's say, when a cell undergoes apoptosis, they form vesicles inside and then it spits out these vesicles. So vesicles are formed. So what's inside these vesicles are undigested substances. So that's how. So like the cell, like it's simply best example is before you move out of house, you pack your stuff and you go out. So just like that, the cell before it's gonna die, it packs its stuff into vesicles and it puts it out. So that spitting process is called exocytosis. So this is basically everything you need to know about cells and transport. And let me know if you guys need any other topic. So drop me in the comment section of anything you need to know and I can make a video on that. And I can make you this short bit. The main reason I'm doing this is because when I was doing my master's, I hate studying. So this is the best way that helped me study. Like, let's say the exam is next week, I just glance at this and figure it. Like, I just need a roadmap and then I'm able to write. So if this helped me, I feel like it's going to help you guys as well. And good luck.